Welcome everyone to the video on microbes and biotechnology. So it's really important that we realize that microorganisms aren't just organisms that we study or try to control. Scientists have actually learned how to harness microbes and the microbial processes they do and use them as tools. So, biotechnology may be a term that you've heard of, but maybe aren't too familiar with. Essentially, what it is it's using biological systems to develop useful products and technologies. So, again, you'll notice bio there at the front of the word because that means we are using living things and their processes in order to develop tools and technologies. Pretty cool. So, biotechnology is going to include kind of like the medicinal world, genetically engineered organisms, which aren't near as scary as people think, diagnostic tools, and new ways of treating diseases. A lot of big biotechnology advances are actually rooted in discoveries that were originally made by studying how bacteria naturally function, right? Bacteria are small but mighty, y'all. Small but mighty. All right, so I have two examples here that we're going to talk about, but please do not think that you are expected to be experts in these processes, okay? Just big picture overview here. Now, if you are interested in them, I highly recommend learning more about them. We'll come back to this a little bit later, um, but really, I just want you to understand that microbes are not just these scary, dangerous, disease-causing, entities. A lot of good has come from studying them. So let's dive in to CRISPR, which has been in the news quite a bit recently. And so CRISPR actually began as this natural defense system used by bacteria to protect themselves against invading bacteriophage. And scientists actually discovered that this microbial system could be adapted into a really powerful tool for making specific targeted changes to DNA. So essentially, y'all, this is allowing researchers to locate and edit specific gene sequences. So far today, CRISPR has applications in biomedical research, agriculture, and the development of treatment for some genetic diseases. So this is pretty cool too, y'all, right? Something that bacteria evolved to defend themselves from viruses has actually become one of our most important tools for studying and potentially treating genetic disease, right? Thank you, bacteria. All right. The next and final topic just for this particular video is bacteria and insulin production. Okay, gotta love bacteria, y'all. All right, so before we had the technological advancements that were needed in order to make recombinant insulin possible, the insulin used to treat diabetes was primarily obtained from the pancreases of pigs and cattle. But scientists learned to place the human gene containing the instructions for insulin into microorganisms like E. coli. And we essentially turned these bacterial cells into tiny insulin producing factories. So bacteria grow really quickly, y'all. They can replicate anywhere from about 20 to 40 minutes, and that's on average. And so, because they can grow quickly, they can produce large amounts of protein quickly that can then be purified for medical use. So, this is a really cool way um, or example of why microbiology matters beyond infectious disease, right? We can use basic machinery of a bacterial prokaryotic cell to make a human protein that millions of people depend on. Okay, you're probably realizing that one of my favorite words is snazzy, but y'all, this is snazzy. Okay. Now, if you are just have had your mind blown and you want to learn more about these, please feel free to reach out. There are some really cool videos on YouTube. There's a lot of other really snazzy resources that I am happy to share with y'all. So please don't hesitate to reach out. But again, for the purpose of any assessments in this class, you don't need to be experts on this, right? You just need to have a basic understanding of what we've talked about so far. All right, y'all. I hope you have a fabulous day. Let me know if you have any questions.